after having shown the role of the thalamus in epilepsy, and before continuing with the hypothalamus and pituitary, I thought it was good to spend a few minutes on the embryology of the deep structures of the forebrain and talk about how the forebrain curves, which is reflected in the shape of the caudate nucleus, and talk about the thalamus and hypothalamus. There are two websites that have helped me to understand the embryology a lot better. The first one is an Australian website by Dr. Mark Hill, and I've also used images from this website in the first Brain Bit by Bit videos on malformations of cortical development. The other website is from the University of Michigan with MR images of human embryos at different Carnegie stages in different directions. And in embryology, I like to start from the point where it was still simple, and that is when the neural plate closes to form the neural tube. This happens at embryonic day 8 in mice and at 3 weeks of human gestation. The anterior neuropore that has closed becomes the lamina terminalis, or the anterior wall of the third ventricle eventually. This is a cast of the ventricular system at embryonic day 9 in mice, 4 weeks human gestation, and you can see the large third ventricle. And from this third ventricle there are two horns, which are the lateral ventricles, that are growing outwards and growing posterior, so expanding the telencephalon. You can see the cerebral hemispheres coming up here. In mice at embryonic day 13, you can recognize some more structures, such as this is again a coronal image, and you can recognize the choroid plexus, the cortical hem, where lots of cells are derived from, among others the glial limiting membrane Cajal-Retzius cells and the hippocampus. And this is a human embryo of 8 weeks, where you can also see the choroid plexus, the lateral ventricles, and two large thalami. This is a human embryo, also 8 weeks, but one Carnegie stage further. And you can see the large lateral ventricles, the third ventricle bordered by the thalami. And if you look at the MR image, which is in the axial plane, you can see that there is very thick neural tissue surrounding the third ventricle, and that the lateral ventricles, so the outgrowth, are limited by a thinner layer of neural tissue. I have colored and outlined the thalamus orange in this picture. Then you can see the basal ganglion, which is also a telencephalic structure, outlined in a purplish color, and the neocortex, which is a telencephalic structure, outlined in red. If you go a little bit more caudally, so I think in red this would be approximately the plane of this histological specimen, you have on both sides of the third ventricle the hypothalamus, and the anterior wall is the lamina terminalis. This is a 12-week-old human fetus, and you can again recognize on ultrasound the thalamus and the lateral ventricles and telencephalic structures, and you can see that the telencephalon cannot grow any more posterior here. So what happens is that after 13 weeks the telencephalon and the cerebral hemispheres start to curve, and you can also see this curving in the cast of the ventricular system in pink, and you can recognize the curving that has happened in the caudate nucleus in adults, that has this weird inverse C shape, and in the hippocampus that was first located in the medial cranial roof of the lateral ventricle, but because of the curvature becomes located in the floor of the lateral ventricle. This is another human fetus of 17 weeks, the thalamus again outlined in orange. And if you would be very dorsal, you would have the choroid plexus, the cortical hem, and the hippocampus here. So at 17 weeks of human gestation, the situation resembles an adult rabbit brain a little bit, where you can also see the lateral ventricle and the hippocampus being located in the cranial medial side. But because of the curvature, the hippocampus ends up medial caudal of the lateral ventricle. You can also see the thalamus again at 24 gestational weeks, which has been divided by the growing fibers from the internal capsule connecting the neocortex and the brain stem into the thalamus and the pallidum. And the same has happened in the striate or basal ganglion, which has been separated by the anterior limb of the internal capsule into a caudate and putamen. The hypothalamus is still located on the floor of the third ventricle, and in the adult situation, the hypothalamus is under or below the thalamus and divided by the hypothalamic sulcus in the third ventricle, and the hypothalamic sulcus runs from the foramen of Monro to the opening of the cerebral aqueduct. So if we would focus on the hypothalamus, and this is a more modern prosomeric model of the embryology, which means that the neural tube is divided into transverse sections, and in each section there's different gene expression, and each section has a different fate. So the hypothalamus is located on the floor of the neural tube. The posterior pituitary is an outgrowth of the hypothalamus. And if we go back to where we started, to the closing of the neural plate, this was becoming the lamina terminalis, and the hypothalamus is now on the floor of the third ventricle, near the optic recess and the infundibular recess, very close to the lamina terminalis, the anterior neuropore. And I imagine that if you go back in evolution to very simple organisms, the hypothalamus is located in the anterior end of the organism on the ventral side, so on the floor where it touches the ground, so it receives all the important information and it makes sure that it has all the functions vital for living and thriving. And that's still the case in humans. And there are many hypothalamic nuclei with a very ingenious network of blood vessels and blood-brain barrier that we will discuss in the future. And the different hypothalamic nuclei cannot be imaged. And I think the description that they are like potatoes in a potato sack is one of the most accurate descriptions that I've read of the hypothalamus and the hypothalamic nuclei. And with this information, I think we can continue in the next video with hypothalamic hamartoma. Thanks